All right, let's dive right in. Today, we're going to strip plant science down to its absolute core and look at a tiny, microscopic marvel that could literally be the key to feeding our future. We're talking about the naked plant cell. And trust me, what it can do is going to blow your mind.
You know, this quote really just hits the nail on the head. We are facing a perfect storm, right? You've got climate change on one side and a global population that's about to hit 10 billion people on the other. Doing things the old way in agriculture, well, that's just not going to cut it anymore.
So let's break down what that pressure actually looks like. Our whole global food system is stretched unbelievably thin, and honestly, in a lot of ways, it is at a breaking point.
Now, the really crazy thing here is how all of these challenges are tangled together. It's a domino effect. Climate change gives us these wild temperature swings and water shortages. That, in turn, wrecks the soil and basically rolls out the red carpet for more pests. Then you throw in pollution and the fact that we have to feed all these people, you can see why scientists are scrambling to find brand new ways to make our crops tougher.
But what if the solution wasn't just a new fertilizer or some fancy farming technique? What if we could completely redesign the plant itself, right from the source code? Well, let's meet the tiny hero of this story, the naked plant cell.
This is the big question, right? This is what's driving a whole revolution in biotechnology. How do we get past the natural limits of breeding to build plants that are just fundamentally stronger, more resilient, and way more productive? Well, the answer starts by taking the plant apart, one cell at a time.
And that brings us right here to this critical definition, a protoplast. It's basically a plant cell that's had its tough outer wall, its armor, really, gently dissolved away. What you're left with is just the cell membrane holding everything inside. Think of it like the living, beating heart of the plant cell, totally exposed and ready for an upgrade.
And here's why that's so important. That cell wall, it's great for protecting the cell out in the wild, but for scientists, it's a huge barrier. By getting rid of it, the cell becomes wide open. New genetic instructions, we're talking DNA, RNA, proteins, they can be slipped right inside, something that's pretty much impossible to do when that wall is intact. This is the gateway.
So how do scientists actually do this? How do they harness the power of these naked cells? Well, this next part is all about the toolkit they use to basically rewrite the rules of plant life.
It all kicks off with this four-step process. First, scientists pick the best material to start with. Younger tissues from leaves or petals work great. Then comes the key move. A cocktail of special enzymes basically acts like a biological solvent, dissolving those cell walls without hurting what's inside. After that, it's pretty straightforward. They filter out the big gunk and use a centrifuge to spin everything down, leaving a pure collection of healthy protoplasts.
This diagram lays out the entire journey perfectly. See over on the left, you start with bits of a plant, like flowers or leaves, and they get broken down. Follow the arrows, and you can see the enzyme treatment, the filtering, the whole 9 yards. This gets scientists a clean batch of protoplasts, ready for action. And from there, they can do all sorts of amazing things. CRISPR gene editing, for example, before, and this is the incredible part, growing that one cell back into a whole new, superpowered plant.
Okay, so we've got our pure batch of naked cells. This is where the real magic happens. Scientists have come up with some seriously powerful ways to get new genetic information inside them. Let's look at the two main methods. So on one hand, you've got chemical fusion. This uses a substance called PEG, which is kind of like a cellular glue. It just sticks the protoplasts together so their insides can mix. It's cheap, it's easy, but it can be a little rough on the cells. But on the other hand, there's electrofusion. This uses a quick electrical zap to open up tiny little pores in the cell membranes. It's super efficient, way gentler, but it needs some pricey, specialized equipment. So, it's a trade-off.
Okay, we've covered the how. Now let's get to the fun part, the why. This is where we see how these tiny, naked cells are being used to engineer a much, much better future.
I mean, just look at the sheer scope of this technology. We're not just talking about making better food. We are talking about turning plants into little factories for vaccines. We're talking about designing plants to literally clean up our planet's pollution. And yeah, we're even talking about using this tech to grow food in space. The potential is just staggering.
For agriculture, this is an absolute game changer. Take something called somatic hybridization. This lets scientists fuse cells from two totally different species, ones that could never breed naturally, to combine their best traits into one superplant. Then you have tools like CRISPR, where they can make these surgical-level edits to a plant's DNA, adding something like drought tolerance with pinpoint accuracy. And what's really amazing is they can often do this without adding any foreign DNA at all, which is a huge deal.
And this isn't sci-fi, it's a concept called plant molecular farming. Forget those giant, sterile, incredibly expensive steel vats. Imagine engineering a simple plant like tobacco to produce crucial proteins for a new vaccine or antibody treatment. You could then grow these life-saving medicines at a massive scale in a field, which could dramatically lower costs and make them available to everyone.
And this technology, it's not just about us, it's for the planet too. Protoplasts give us a green, living solution to our pollution problems. Scientists can literally design plants with a supercharged ability to suck up toxic heavy metals from the ground, or to break down nasty pesticides in the soil. It's like sending in a botanical cleanup crew, using nature itself to heal the damage we've done.
And hey, as we start looking toward the stars, this tech might just be our ticket there. For any long-term mission to Mars or the Moon, you just can't pack enough sandwiches. Protoplasts are the key to building sustainable farms off-world. They let us grow food incredibly efficiently, without soil, in the tightly controlled environment of a spaceship or a Mars colony. It's a level of adaptation that a normal plant could never achieve.
So where does all this lead? Let's zoom out for a second and really look at the path forward and the almost unbelievable power that's locked inside just a single one of these cells. So, to boil it all down, protoplasts are the master key that unlocks a plant's genetic code. They let scientists break the rules of nature, creating powerful new hybrids. They make genetic editing fast, precise, and clean. And, as we've seen, the applications are transforming everything from the food on our plates and the medicine in our cabinets to the future of how we explore the universe.
And that leaves us with this one big, thought-provoking question. We've seen that a single, humble plant cell, once you strip it bare, holds this almost limitless potential to solve some of our biggest challenges. So the question for all of us is, what should we build with it? Thanks for tuning in.